Hey everyone, today we're going to be detectives. We're going to investigate one of the biggest mysteries in biology. How in the world do plants actually make their own food? We're going to look at the amazing experiments that cracked this code.
I mean, it sounds like a pretty simple question, doesn't it? But think about it. For hundreds of years, this was a massive puzzle. You see a tiny little seed turn into a giant tree, and it seems to happen with just a bit of soil and water, almost like magic. So what are the real ingredients? That's the big question we're diving into.
And you know what the best part is? We're not just going to learn the answer. Nope. We're going to walk in the footsteps of the scientists who figured it all out. We'll break down their super clever experiments step by step, so you can see the evidence for yourself.
Okay, to make sure you absolutely nail this for your exams, here's our game plan. First, we'll prove that leaves actually make food. Then, we'll get into the history and solve the big puzzle. After that, we'll tackle some real board exam questions and we'll wrap it all up with a super fast revision. Let's do this.
All right, first things first. Before we can figure out how plants make food, we need solid proof that they do make food, right? The food they make and store is a carbohydrate called starch, and lucky for us, there's a really cool chemical test to find it.
So let's break down this procedure, because every single step is there for a smart reason. We boil it in water first to kill the cells and just make the leaf a bit softer. Then, and this is the crucial part, we boil it in methylated spirit. Why? To get rid of all that green stuff, the chlorophyll, which would totally hide our result. After that, we just give it a quick wash and add the magic ingredient, iodine.
And boom, there it is, the big aha moment. Seeing that pale, washed-out leaf suddenly turn this deep, bluish-black color, well, that's our definitive proof. It's a chemical signal that screams, yup, starch is here. No doubt about it.
So this brings us to a key definition, and you'll want to star this in your notes because it's a classic exam question. The starch test is simply a chemical test where iodine solution, which is usually brownish, reacts with starch and turns bluish-black. Simple, direct, and you absolutely need to remember it. Okay, moving on to the next part of our investigation. We know leaves make starch. Great. But how did scientists figure out what raw materials, what ingredients plants even need for this whole process?
For this, we have to travel way back to the 1770s and meet a scientist named Joseph Priestley. What's so amazing is that he did all of this before anyone even knew oxygen existed. He was working with a very simple setup: a sealed glass jar, a candle, a mouse, and a little mint plant.
So first, Priestley identified the problem. He lit a candle, put the jar over it, and poof, the flame went out. He put a mouse under the jar, and pretty soon the poor thing suffocated. He concluded that both the burning candle and the breathing mouse were doing something to the air, something that damaged it. They were using up some kind of essential stuff in the air.
And this is where it gets really cool. He tried the experiments again, but this time he stuck a mint plant in the jar with them. And guess what? With the plant in there, the candle kept on burning. And the mouse stayed alive. The plant was somehow doing the exact opposite. It was restoring the air that the candle and the mouse had damaged.
This simple sentence was his final hypothesis, and it was absolutely revolutionary. For the very first time, someone had shown with an experiment that plants and animals have opposite effects on the air. They create a kind of balance. This was huge. It proved that some kind of gas exchange was happening and that plants were releasing something that supports life.
All right, theory's done. Let's put this knowledge to the test. Now we're going to walk through exactly how these ideas show up on your SSC board exams. We'll break down a few common questions so you know how to write those perfect full-mark answers.
Okay, let's kick things off with a 1-marker and a 2-marker. First question, what is the role of methylated spirit in the starch test? And second, what did Priestley conclude from his bell jar experiments? Go ahead and pause here, think about your answers.
So here's what the examiner is looking for. For that 1 mark, you have to say that the spirit removes chlorophyll to make the result visible. Just saying it cleans the leaf isn't enough. For the 2 marks on Priestley, you have to mention both sides of the coin, that animals damage the air and that plants restore it.
All right, let's level up to a 3-mark question. This one is super common, asking you to describe the procedure for the starch test. Can you list out those key steps in the right order?
To get full marks, you need to list those steps clearly. Now here's a pro tip. A really common mistake students make. They forget to mention the water bath. You have to remember methylated spirit is flammable. You can't heat it directly over a flame. You must heat it indirectly, in a test tube placed in a beaker of boiling water. That one little detail shows the examiner you really know your stuff.
And now for the big one, a 5-mark long answer question. This is where you have to tell the complete story of Priestley's experiment. You can't just list facts, you have to connect the dots and build a logical argument showing how he reached his conclusion.
To nail this 5-marker, you need all these parts. Start with his first experiments, the control group, where the air gets damaged. Then, describe the main event with the plant, where the air gets restored. Finally, state the big conclusion about gaseous exchange and wrap it up with his exact hypothesis. You have to show both parts to tell the full story.
Our last practice question is all about drawing. In biology, a good, clean, well-labeled diagram is worth its weight in gold. So let's think about that setup for getting the chlorophyll out of the leaf. What are the essential things you need to draw and label?
Okay, these are the labels you just can't miss. Your marks are for 2 things, drawing it neatly and getting these labels right. And I'm going to say it one more time because it is so important. Your drawing must show the test tube inside the beaker of water. That's how you show the water bath. Don't even think about drawing that test tube directly over the flame.
Fantastic work, everyone. To make sure all of this is locked into your brain for good, we're going to do a super fast 60-second rapid revision of all the most important points. You ready? Let's go.
Okay, let's fly through it. The starch test proves food is in leaves. The chemical you use is iodine solution. A positive result is a bluish-black color. To see it, you got to remove the green chlorophyll with methylated spirit, and you do that safely in a water bath. What did Priestley find? Plants restore the air. His big conclusion? Plants perform a gaseous exchange that's essential for life.
And we'll end with this final thought. Priestley proved that plants restore the air, but he had no idea which specific gases were involved. His amazing work set the stage for the next group of scientists to figure that out and eventually discover oxygen. But that's a story for another time. Thanks for joining me on this investigation.